Welcome to this demonstration video of proton transfer mass spectrometry, or PTR-MS. My name is Sylvester Holt and I'm an assistant professor at the Department of Food Science at University of Copenhagen. We have purchased the Onicon PTR-TOF 6000 with funding from FOODHAY. FOODHAY is the Food and Health Open Laboratory with a national initiative that is funded by the Ministry of Higher Education and Sciences. Applications of PTR-MS are very broad in food science, and new applications are added every year. Some applications are listed below. Real-time breath analysis during food consumption. Dynamic monitoring of formation of molecules during food processing of many different types of food. Quality control of dairy products. Monitoring of off flavors. Headspace fingerprinting of food products without target analysis. This can be used to differentiate products without monitoring target compounds, but rather looking at the entire profile and making correlations to the attributes. When an autosampler is installed, it is possible to make high throughput screening due to the very short time of analysis. Apart from these applications, the PTR-MS is also used for medical applications, environmental research, screening for explosives of chemical weapons in airports, and many more. We will now show you 3 applications of the PTR-MS machine that we have chosen based on ongoing research in our lab. We will showcase how the nose-based sampling works by drinking a cup of coffee. We will show you an example of how we can monitor the coffee volatiles during a roasting test. And finally, we will show you how the PTR-MS can be used for fast measurements by combining it with an autosampler. Here we have the Onicon PTR-TOF 6000 proton transfer reaction mass spectrometer. The PTR-MS is an online technique that can record a spectrum in less than 100 milliseconds with a sensitivity of parts per trillion. That is 1 molecule of analyte per trillion molecules of air. This machine allows immediate detection of differences in the composition of the gas that you are measuring. Inside this box, there's a reservoir for water that is used to generate the charged hydronium ions for the proton transfer reaction, the PTR, that occurs to ionize the molecules in the sample gas that you are measuring. The charged gas molecules are then accelerated and detected in a time-of-flight TOF mass spectrometer. Time-of-flight detectors are the most popular detectors for PTR-MS because they provide a good separation of the masses. The mass resolution of this machine is higher than 6000, meaning that a mass over peak width of 6000 or more. For example, a molecule with molecular weight of 150 will have a peak width of 0.025. This is a NASA module where you place your mouth in front and guide the 2 plastic tubes into your nostrils to measure the gas exhaust from your nose. When you're eating food or drinking a beverage, aroma compounds are evaporating in your mouth to different degrees depending on many factors, such as enzymes in the saliva, pH, oral microbiota, etc. This means that every person has the unique fingerprint of volatile compounds that can be measured. Now let's see what happens when I drink a cup of coffee and breathe into the nose-based sampler. This time you could see that several coffee volatiles were increased in the breath. Notice how multiple additional peaks in the spectra appear originating from the coffee.
Hi everyone, my name is Andika. I'm a PhD student at the Department of Food Science. I'm working with authentication of pea berry coffee. Coupling coffee roasting process with PTR-MS will help us to understand the formation of the desired aroma during an exothermic pyrolysis reaction. We are currently in the process of setting up monitoring coffee roasting volatiles from this coffee roaster.
My name is Pedro and I'm a PhD student here at the Department of Food Science of the University of Copenhagen. Here today I would like to show you an example of a small experiment we have done to analyze thiols and sulfites with the PTR-TOF MS. The setup we have used is the following. An autosampler connected to a GC oven, which is at the same time connected to a PTR-MS transfer line through the GC injection port. Thanks to this setup, we could create long sequences of samples that are injected every 2 or 3 minutes. So this would depend on the incubation time necessary, of course.
Thank you for spending a few minutes with us and the PTR-MS machine. If you would like to use the equipment for your research or would like to know more about the applications, please do not hesitate to contact us by email.